Hello, everyone. Welcome back. In the previous lecture, we learned that adaptive immunity differs from innate immunity because it recognizes specific antigens and can generate long-term memory. But how does the immune system accomplish this remarkable specificity? In this lecture, we'll meet the major cells of adaptive immunity, B cells and T cells, and learn how they generate targeted responses through a process known as clonal selection. By the end of this module, you should be able to differentiate between B and T cells, explain clonal selection and clonal expansion, and describe how adaptive immunity generates specific responses to antigens. Adaptive immunity is carried out primarily by a group of white blood cells called lymphocytes. The two major types of lymphocytes are B cells and T cells. Although both participate in adaptive immunity, they perform different functions. B cells are responsible for producing antibodies, while T cells coordinate immune responses and directly eliminate infected cells. Together, these two cell types allow the immune system to recognize and respond to an enormous variety of pathogens. B cells develop and mature in the bone marrow, which is where the B in B cell originates. The primary role of B cells is to recognize antigens and produce antibodies. When activated, B cells divide and differentiate into plasma cells, which are specialized antigen antibody producing factories. Pardon me. Some activated B cells also become memory B cells. These cells can persist long after an infection has been cleared and contribute to faster responses during future encounters with the same pathogen. We'll explore antibodies in much greater detail in a later lecture. T cells mature in the thymus, which is why they are called T cells. Unlike B cells, T cells do not produce antibodies. Instead, they perform a variety of cellular functions that helps control infections. Some T cells coordinate and regulate immune responses, while others directly kill infected cells. Together, T cells play a critical role in defending the body against intracellular pathogens such as viruses. We'll learn more about the different types of T cells and how they are activated in the next lecture. One of the most remarkable features of adaptive immunity is that every lymphocyte is unique. Each B cell carries a unique B cell receptor, or BCR, while each T cell carries a unique T cell receptor, or TCR. These receptors differ from cell to cell, creating millions of potential antigen specificities throughout the entire body. Before an infection ever occurs, your immune system already contains an enormous library of lymphocytes capable of recognizing many different antigens. Most of these cells will never encounter the antigen they recognize. However, this diversity ensures that at least a few lymphocytes are likely to recognize almost any pathogen the body encounters. So if millions of different lymphocytes already exist, how does the immune system know which ones to activate? The answer is a process called clonal selection. Each lymphocyte recognizes only one specific epitope. When a pathogen enters the body, only the lymphocyte whose receptors match the epitope will become activated. All of the other lymphocytes remain inactive. In other words, the antigen selects the lymphocyte that can recognize it. This is why the process is called clonal selection. Once a matching lymphocyte is selected, the immune system needs more than just one cell to fight the infection. The activated lymphocyte begins dividing rapidly through a process known as clonal expansion. A single activated cell can produce thousands of genetically identical descendants. Because all of these cells originated from the same parent cell, they all recognize the same antigen. This rapid increase in cell number allows the immune system to generate a strong and highly specific response against the invading pathogen. After clonal expansion, the newly produced cells can follow two major paths. Some become effector cells. Effector cells actively participate in eliminating the current infection. Plasma cells produce antibodies, helper T cells coordinate immune responses, and cytotoxic T cells destroy infected cells. Others become memory cells. Memory cells typically remain in the body for months, years, or even decades after infection has been cleared. These cells do not actively fight infection unless the pathogen is encountered again. However, if re-exposure occurs, memory cells respond much more rapidly and effectively than naive lymphocytes. This ability to remember previous infections is the foundation of long-term immunity and vaccination. So, in review, adaptive immunity is carried out primarily by B cells and T cells. B cells produce antibodies, while T cells coordinate immune responses and eliminate infected cells. Each lymphocyte possesses a unique receptor capable of recognizing a specific epitope. When a matching antigen is encountered, that lymphocyte is selected and activated through clonal selection. Activated lymphocytes then undergo clonal expansion, producing large numbers of identical cells that can respond to the pathogen. Some of these cells become effector cells that combat the current infection, while others become memory cells that provide long-term protection. In the next lecture, we'll examine how antigens are presented to T cells and explore the critical role of MHC molecules in adaptive immunity. With that, thanks for attending this lecture, and I'll see you in the next one.